So now we are going to take a closer look at a eukaryotic cell. So we're talking about animal cells and plant cells here for the most part. And we're going to look at the specific organelles within this cell. So if I kind of zoom in here really quickly, this is kind of the general structure of an animal cell here. And we're going to take a look at each of these different organelles that are labeled here and talk about them in a little bit more detail. So first, what we're going to do is start with what's called the plasma membrane. And we've already talked briefly about the plasma membrane and of phospholipids, and we know that it's kind of the barrier to the cell. So we can see the plasma membrane here. It's this labeled, it's this purple, it's colored here, this purple area here. That's the outside of the cell. So when we talk about the plasma membrane, what we are talking about, right, so we'll talk about the different functions of the plasma membrane. So when we talk about the functions, we already talked about them briefly. One, it contains the cell and it separates it from the environment. So it contains the cell and separates it from the environment. So again, this is kind of a wall, kind of a barrier. This is where the cell starts and or ends. And so then we have this kind of separation between it and the thing outside of it. So the fluid outside of it, the other cells outside of it. And it contains all the stuff inside of it, so it wants to keep all of its insides in the inside. Secondly, it regulates what goes in and out of the cell. So it regulates what goes in and out of the cell. Plasma membrane, and we're going to talk about it in even more detail, but the plasma membrane is fluid. It allows things to come into the cell. When we want things to come into the cell, we want things to leave the cell, like waste. So we want nutrients to come into the cell, like glucose or sodium or chloride or calcium, different ions and things to come into the cell so that we can use them, break them down, use them for energy, for example, with glucose. We also want things to leave the cell. So things like carbon dioxide, we want to leave the cell. That's a waste product. We want uric acid or urea to leave the cell. So we can regulate what goes in and out of the cell. Other things we don't want to leave the cell. So we have our amino acids that are floating around in the cytosol. We don't want our amino acids to leave the cell, so it's going to contain those things, keep them inside. Also, we want to regulate what doesn't get in the cell, right? So hopefully something like bacteria doesn't get in the cell. So we have the plasma membrane that's going to try to protect itself, protect the cell from allowing things like bacteria in or viruses in. And then third, the third function of the plasma membrane we'll talk about right now is that it interacts with other cells and the environment. So our cells communicate. We do have very specific communication cells like neurons in our body that are communication cells within our brain and our spinal cord and throughout the body and our nerves. But we also have communication in other ways. So those that are able to secrete hormones, for example, for communication. So that's one way to interact with other cells. We have the ability to have little channels that go between the cells that are within the plasma membrane that allow things to move between cells, and allows it to interact with the environment in that same way. So we have little channels that allow things to get into the cell, allow things to get out of the cell. We also have different finger-like projections on the cell, like microvilli or cilia. Cilia are on top of the cell that are going to be moving things across the cell, kind of like cilia are in the lungs, for example. When we're mucousy, we cough out things. The cilia in the cells or on the cells in our lungs are helping to get that out. Those are part of the plasma membrane that allows it to interact with other cells and with the environment. So, for example, with the mucus and coughing things out, that's our cells interacting with the environment, or the plasma membrane specifically interacting with the environment. So let's take a look at the structure of the plasma membrane. So the structure is said to be a semi-permeable fluid mosaic model. So it's semi-permeable fluid mosaic model. So the semi-permeable piece we're going to talk about in more detail a little bit later. But semi meaning somewhat, and then permeable meaning allows some things in and does not allow other things in. So allows some things in and does not allow other things in. So permeable, meaning allowing things to come through it, and it's somewhat permeable. Doesn't allow everything in, otherwise it would be completely permeable. Or if it was not permeable at all, then it would let nothing in. So it's semi-permeable, allows some things in, some things not. Now this fluid mosaic model piece means that it is fluid, meaning that the pieces, the parts that make up the plasma membrane are flexible. They're moving around. They can change shape. So let's put fluid here. So fluid meaning the membrane is flexible. And can change shape. Oops. Wow. And can change shape. So I'm going to kind of draw our membrane here for a moment. What I'm drawing right here is a very, very shorthand. If you recall, in our biological molecules, our phospholipid, right, was the main component of our plasma membranes. Remember our phospholipid here, right, we have our head and tails, and our head was polar, and our tails were non-polar. And remember, they kind of sequestered themselves. So here, I'm pretending that my kind of squiggly or swirly part at the top are a whole bunch of phospholipid heads. That's, again, my shorthand, but they would be individual circles. And then these squiggly lines at the bottom are the tails. Again, very, very shorthand. There are 2 tails per single head. But to get the idea, I'm just kind of drawing it very, very quickly like this. And these are just the phospholipids. So this is the main structure of our plasma membrane here. The main structure is that it is a phospholipid bilayer. Bi meaning 2, so 2 layers of phospholipids. We can see that here, 1 layer, 2 layers. And again, out here, this would be a fluid area, and this would be a fluid area. So this would be, say, inside the cell. And we already talked about what was inside of the cell. We have this watery, liquidy gel mixture called the cytosol, or the cytoplasm. And then outside of the cell is also fluidy. We have extracellular fluid. Extra meaning outside of, and then cellular, outside of the cell fluid. So we have the fluid inside of the cell, and we have the extracellular fluid, the fluid outside of the cell. So we have our phospholipid bilayer that is the main component, or those are the main components of our plasma membrane. Now, this fluidity is due to the fact that these molecules, these phospholipids, can move around inside of the membrane. So this one can move over here, this one can move over here. They can kind of all shift around. If you think about putting putting some beads in water, if you dump enough beads to cover some water and you kind of moved the container around, let's say that you put a tub, or you fill a tub with some water at the bottom and you dump a whole bunch of beads in there, little round beads. And then you kind of slosh the water around gently, not huge waves, but you gently slosh it around. You can see that the beads will be moving around as they're floating on the surface of the water. They're still kind of maintaining a layer on top of the water, but they can move around within that layer that they're still maintaining. And this is what we see with the phospholipids. You can kind of imagine that this head, the polar head, would be like the beads sitting on top of the water, where they're maintaining this layer, but they're able to move within it. And then it can also become, it can change shape, meaning that the cell itself can stretch out and get smaller. There are some cells that we'll talk about called macrophages that create pseudopods, which are fake feet. And so they kind of plop out some of the cytoplasm into kind of this long arm type shape, and then they fill that arm shape with the cytoplasm, moving it forward, and then kind of plop out more cytoplasm and then move that forward. So it can actually change the shape of the cell itself. So part of this fluidity is, as we mentioned, that the molecules of the plasma membrane can move around within the membrane. And then also we have different pieces to the plasma membrane. So we have the phospholipids, which makes up the majority of the plasma membrane. But we did also mention that we have cholesterol inside of the plasma membrane. Remember, cholesterol is one of our sterols, a 4-ringed sterol. I'm kind of generally drawing these 4 rings in here. And it actually acts as a way to keep our membrane fluid. And again, that's because it's going to create spaces that these phospholipids can move around. So rather than packing in really close to each other, they then can be moving around. It's hard to do this, but it would be more like this. Moving around this large chunk of cholesterol. And that space is going to allow it to be more fluid. And then also, not only do we have, the cholesterol, the unsaturated fatty acids. So remember, our unsaturated fatty acids allow for space. Our unsaturated fatty acids, we have lipids in here that are going to be connected to glucose chains out here, which we'll talk more about. And those lipids can have unsaturated fatty acids. And those unsaturated fatty acids will make it even more fluid, so we don't have that packed-in effect that we saw with our saturated fatty acids. Now that covers semi-permeable, and that covers fluid. Now the last piece of this is mosaic. And the reason that it's called a mosaic is because there are many molecules in the membrane. So if you know what a mosaic is typically a piece of artwork or something that is made up of a whole bunch of little tiny pieces. So a lot of times this will be done with tile, for example, lots of different kinds of tile, and then you put them all together, these little tiny pieces, and then when you step back, you can see this whole big picture. If you look really, really close, all you might see are a couple of different shaped or different colored tiles. And so a mosaic is used to describe the plasma membrane. Because there are different molecules in here. So not only are there all of these individual phospholipids that if you look really close, you're just looking at those phospholipids, but then there are also things like cholesterol in there. We also have different proteins that we'll talk about being embedded in there. We also have different things called glycolipids, and we have glycoproteins that are in there. And so we have different types of molecules in the plasma membrane. We're going to go through those in just a moment. And then we also have many molecules in there. So that's where we get the mosaic piece from. So it's a semi-permeable fluid mosaic model. So then the last thing I want to say about the plasma membrane right now, and we're going to get into a lot more detail about the plasma membrane in another lecture, is when we talk about the many molecules of the membrane. Actually, I think I'll do it over here. Many molecules of the membrane. First of all, we're talking about the phospholipids, which we know make up the majority of the membrane. Then we also have the cholesterol, which we've talked about. And the cholesterol is going to keep the membrane fluidity. Then we also have glycoproteins. And our glycoproteins are, as the name implies, proteins with carbohydrates attached. So that's the glyco part. Glyco meaning glycogen. So glycogen and proteins. So proteins with carbohydrate chains attached to them. We also have glycolipids, as their name implies, lipids with carbohydrate chains attached. And then we have membrane proteins. And we have two different types of proteins to mention. We have what are called integral proteins. And those are proteins that go all the way through the bilayers. So when we're looking at the plasma membrane, and we have our tails, our heads, we can have a protein that's going to go all the way through the two layers here. So this would be an integral protein. Then we also have our peripheral proteins. So these are proteins that are associated with the membrane. They don't go all the way through. So we would see a peripheral membrane just on one side of the membrane here. And then we would still see our phospholipids, for example, over here. Okay. So then I can kind of draw in our cholesterol, our 4 sterile ringed molecule, which gives fluidity. Oops. Make that phospholipid bigger. And then I can even draw in one of our glycoproteins. So if we did a glycoprotein, we would have a protein here, kind of drawing over my words here, but a protein, and then we would have our chain of carbohydrates coming off of the top of the glycoprotein. And then similarly, let me erase some over here maybe. I don't know, we'll throw it above the words again. So we have more phospholipids, but then we would have our glycolipid. And so our glycolipid would be some unsaturated fatty acids that have glycogen chains attached as well, meaning carbohydrates. So here, this is our glycoprotein, and here, glycolipid. So you can probably do a better job than me if you have some more space here. I kind of started running over my words. But we have the phospholipids, which are the majority. We have our cholesterol, our 4 sterile ring cholesterol, our glycoproteins, glycolipids, and then our different types of proteins. So right now, that's what I want to go over for the plasma membrane. We're going to talk a lot more about the plasma membrane at a later time and how we're able to utilize this semi-permeability. So for a moment, what I would like to talk about, which is not in this actual drawing, would be the cell wall, because I do want to address the cell wall as it relates to plants. So again, plants have a cell wall. Do that in green. So the cell wall for plants, we have it in plants and in fungi. Not in animal cells. So none of our body cells have a cell wall. So the function of the cell wall is that it protects the cell. It helps to maintain its shape, and then it prevents it from bursting under pressure. So protects the cell, helps maintain shape of the cell, and it prevents it from bursting. And the structure of the cell wall, we have talked about a little bit already, but it is a thick wall that is very, very rigid, which is, of course, why it helps to maintain its shape and prevents it from bursting. And it's made up of either cellulose in or chitin in fungi. And the cell wall is around the plasma membrane. So we would typically see something like a plant cell where we have the plasma membrane here. Plant cell has a nucleus. We'll put the nucleus in there. And then we would have the cell wall kind of around the plasma membrane. And so there can be a little bit of space between the plasma membrane and the cell wall of the plant cell, and that's because sometimes we'll have water move into the cell, and then the plasma membrane will get even closer and fill up all of the space in that cell, within that cell wall. But then, of course, the cell wall prevents it from actually bursting, from breaking apart. And then also we can have water be leaving. If a plant doesn't get watered, then the plasma membrane can shrink down, and then it'll get smaller, and then it won't take up as much space within the cell wall. But the cell wall will stay out there, maintaining the structure of the plant. So the plant itself doesn't actually shrink down or shrivel up, per se, because it still has that cell wall out there maintaining the shape of the cell. All right, so just a quick word about the cell wall. So now let's move back to our image of our eukaryotic cell. We've talked about the plasma membrane. Now what we've mentioned several times already is the nucleus. So I'd like to talk about the nucleus. So the nucleus is right up here. Well, the name for the nucleus, or the label for the nucleus, is right up here. And what we are talking about here, then, is this envelope. You can see where it's pointing is kind of this orange, holy structure. So this is an additional plasma membrane that's within the cell itself. So you can see it's kind of a ball. And so we have a circular plasma membrane, and there's the empty space in the middle, not actually empty, but there's space in the middle that is where the DNA is contained. So this whole thing is the nucleus. It includes the nuclear envelope that you can see labeled here. The nuclear envelope is the actual outside portion here. The actual plasma membrane that has the holes in it. And then we have our nucleolus, which is where we have a region of ribosomes and DNA. And then we have the chromatin, which is part of our DNA, is the chromosomes. And we'll talk more about that as well. So why don't we go ahead and kind of zoom into the nucleus here. So when we're talking about the nucleus, let's first talk about the function. So a lot of times when people talk about the nucleus, they talk about it as the headquarters of the cell. So the headquarters of the cell, meaning that's where all of the information is coming out of. Remember, it maintains the information in our DNA, and it sends that information out. So the function of the nucleus then is to store and protect the DNA, and then this is where we have DNA and RNA synthesis. So store and protect DNA. And then this is where we have, this is the site of DNA and RNA synthesis. The structure, we already mentioned. The structure, we have our envelope, which is a double membrane. So just like our plasma membrane is a bilayer, we have a double membrane around the nucleus. We have the nuclear pores. So those were the holes in the membrane. That allows for the RNA to leave the nucleus and get into the cytosol. Allows things to get in and out. And then we have the nucleolus. The nucleolus is this dense area that stores proteins and RNA to make ribosomes. So I kind of shorthanded earlier, and I said that's where ribosomes are, but that's where the information is for ribosomes. So proteins and RNA that are going to make ribosomes, and the ribosomes are actually in the cytosol. So in the nucleus, again, we have our DNA And remember our DNA, our deoxyribonucleic acid. Remember this contains the instructions. Wow, that's messy. Let's try that again. So the instructions for how to make all the proteins. For that cell. So all the proteins for the cell. Now remember its structure, it's a double helix. We have our nucleic acids. They have our 4 bases, the A, T, C, and G. And then it's packaged to fit into the cell. So it's really, really long strands of this, but we actually take our really long strands of DNA and we wrap it, so it's wrapped around proteins called histones. So histones are kind of like those little things that hold the thread. So if you think about if we take thread, or if we take yarn, let's say, we have an entire skein of yarn. This yarn is all over the place. If you think of a yarn ball, but it's all over the floor, let's say, that would be like our DNA. Our DNA can hang out in the nucleus just all open, and the reason it would be open like that, see if I kind of zoom in here, you can kind of see that it's squiggly all over the place inside of the nucleus here. And that's when the yarn is all over the floor. But then what we can do in order to make it nice and easy and compact is we can take these proteins called histones and we can take the DNA and we can wrap them around the histones. And then, instead of having this long DNA yarn all over the floor, then if we have these different proteins called histones, we can make them nice and neat and keep wrapping the DNA around these histones. And then what we can do is we can take these histones and wrap the histones that are covered in DNA around the histones. And then what that does is, you can see, it makes it even more compact. And then we can take our big clumps of histones and wrap them around again and again and again. And when we get them really, really, really tiny and compact as much as we can, they end up looking something like this. This is actually a duplicated chromosome. Oftentimes when we do karyotype, which is when we're looking at chromosomes, they're duplicated. Actually, though, when we're talking about non-duplicated or non-copied chromosomes, we're talking about individual chromosomes that look something like this. We'll get into that in more detail when we get to cell processes. What's important to know right now is that our DNA is really, really long. It's about 2 meters. 2 meters long, and then we take that and we wrap it around these different proteins in order to make sure that it all stays neat and tidy, we don't get any knots in it, and then we can open up sections when we want to open up sections, and we can then go through processes like cell duplication, which is mitosis, or we can go through meiosis as well, where we're going to condense this information and separate it out and make copies of it, and it's important that we be able to do that. All right, so the next organelle that I'd like to talk about, I'm actually going to go back over here by our cell wall, and that's because it's the chloroplast. I just want to mention very briefly the chloroplast, which is, again, not part of an animal cell, but is only part of a plant cell. So, plant cell only. So the chloroplast, the function of the chloroplast is to use light energy to convert carbon dioxide and water into glucose and oxygen. Use light energy to convert carbon dioxide and water into glucose and oxygen. So our photosynthesis is what's happening here in the chloroplast. So this is where we're taking in light energy, or plants rather, are taking in light energy, and they're going through photosynthesis. So really quickly, our equation for photosynthesis is 6 carbon dioxide plus 6 water is going to be converted into glucose, C6H12O6, and 6 oxygen. So you should know the equation for photosynthesis. We're not going to go into too much detail, just to say that it is an anabolic reaction. So we're taking these smaller molecules, carbon dioxide and water, and converting it into a more complex molecule like glucose, and then the byproduct of that is oxygen. For plants, their waste product is oxygen. So they have carbon dioxide, they want carbon dioxide, they want water, and what they make is glucose, and then their waste product here is oxygen. And that's kind of strange because in humans, what we think of as our waste product, what we're breathing out, is carbon dioxide. But of course, that's why this all works very well here on Earth, is that what we want is glucose and oxygen, and what we then get rid of is carbon dioxide. And we also get rid of water, even though we want water as well. So this is an anabolic reaction, which means that it requires energy. Fortunately, they're getting a lot of energy from light, from the sun. So that's really all we're going to talk about when we talk about chloroplasts, or when we talk about plant cells. There, the cell wall and then the chloroplast. So that's making energy for a plant cell. The organelle that's making energy for an animal cell is down here, the mitochondrion. Mitochondrion is singular, mitochondria is plural. So you can see they're kind of this bean-shaped organelle. So let's go ahead and take a look at the mitochondria. So our mitochondria oftentimes is called the powerhouse of the cell. So the powerhouse of the cell, or the power plant of the cell. And as its name implies, its function then is to harvest energy from glucose and use it to make ATP. So remember, ATP is our energy storage molecule. What we do is we take glucose, we break it down, we get the energy from that glucose, and we put it into molecules of ATP. Then we can use those molecules of ATP anywhere that we want to in the cell. So we get the energy from glucose. Say we eat a plant, we get it from that glucose they just made, we breathe in that oxygen, and then we convert it into ATP that we can use wherever we want to in the cell. This process is called cellular respiration. And the equation for cellular respiration is C6H12O6, because of course we're talking about glucose, plus oxygen. That's going to go to carbon dioxide and water and ATP. So if you'll notice, this looks about the same as photosynthesis, except backwards. So again, we utilize the glucose and the oxygen, we turn it into carbon dioxide, which we breathe out, and water, and ATP. So the structure then of our mitochondria, so again, our mitochondria is going through this cellular respiration and getting us lots and lots and lots of energy over here in the form of ATP, which is our high energy molecule. So the structure is that we have an outer membrane that surrounds the mitochondria, and then we have an inner membrane. So the outer membrane is keeping the mitochondria separate from its surroundings. So it's separating it, making it an organelle. And then we have the inner membrane. And the inner membrane has lots and lots of folds, which gives it a high amount of surface area. These folds are called cristae. And so the reason that this has a high amount of surface area is because what is found on this inner membrane are proteins that are needed to make ATP. So the reason that we have all of this inner membrane and it's so highly folded and we have an increased surface area is because all throughout this plasma membrane, which is a plasma membrane, this inner membrane, are these proteins that are actually going about the business of making the ATP. So if we kind of look at the illustration again of the mitochondria, you can see here that we have this outer membrane, and then you can see this folded inner membrane. And all throughout this inner membrane, we have these proteins that are studding the whole membrane all along here. And all of these proteins in these cristae, these folded regions, are what are going to be contributing to making ATP. So taking the glucose and breaking it down and getting it into ATP. So taking a look at our cell again, let's move on to our next organelle, which is actually not much of an organelle, but it's called a ribosome here. So a free ribosome, you can see here, there's a free ribosome, but then you can also see up here in the upper right-hand corner, we have a ribosome. And that's because our ribosomes can be found on our rough endoplasmic reticulum, but they can also be free ribosomes, as are indicated in the bottom left-hand corner. So they can float around in the cytosol or the cytoplasm, but they can also be attached to the rough endoplasmic reticulum. So why don't we go ahead and we'll zoom in on our free ribosome here. So when we're talking about our ribosome, these are the workers of the cell. And the reason that they're the workers is because the function of the ribosomes is protein synthesis. So protein synthesis, this is where we have our RNA. Remember, our DNA is what contains the information for making proteins. In the nucleus, our DNA is going to make a copy of itself, a rough copy called RNA. It's not an exact copy because we have uracil in RNA instead of thymine. So we have our single-stranded RNA that's made in the nucleus. That leaves the nucleus, goes out into the cytoplasm where it meets up with a ribosome, whether it's a free ribosome or one that's attached to the rough endoplasmic reticulum. Once it does that, then it's going to contribute to making proteins. So it has the information for how to make a protein, that specific protein, and it uses that information to work with the ribosome to make the protein, utilizing amino acids as well. So we have protein synthesis is the function of the ribosome. It binds to the mRNA, remember the messenger RNA, and it joins the correct amino acid to make proteins. So it's going to combine, it's going to make those peptide bonds. Remember we talked about amino acids, they're coming together through peptide bonds to make polypeptides. So our ribosomes are going to be assisting with that. Our ribosomes come together with our mRNA, and then they're going to attach the amino acids to each other via peptide bonds to make our polypeptides. So the structure, then, of our ribosome is that it's made up of our RNA. Remember when we looked at our 3 different types of RNA, one of them was ribosomal RNA. So our RNA is ribosomal RNA and proteins. So you can see kind of in the illustration here, it looks just like a little tiny dot thing. And that's because it really is just kind of a lump of different proteins with our RNA in there. And then our mRNA comes along and will be in there bringing our individual amino acids together through a process that we'll talk about when we talk about our cellular processes. So our ribosomes, again, are the workers of the cell. They are the ones that make proteins in the cell. And remember, proteins are extremely important. They do so many things in the body. That's what mostly everything does in the body. So most of the things in our body are proteins, and that's because they do so much in the body. We talked about those functions. So the next one that we'll talk about is the rough endoplasmic reticulum. And so we see that the ribosomes are on the rough endoplasmic reticulum. The rough endoplasmic reticulum is kind of this folded plasma membrane. So we'll zoom in here and we'll talk about our rough endoplasmic reticulum. So we actually have 2 different endoplasmic reticulums. We have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. First, we'll talk about the rough. The rough endoplasmic reticulum is seen as a factory. So our factory, because when we talk about the rough endoplasmic reticulum, we have these ribosomes, right? So they have ribosomes attached to, let's see, this would be the structure. So the structure of our rough endoplasmic reticulum is that they have ribosomes that are attached to a series of membrane channels. So we saw that they were folded plasma membranes. So ribosomes attached to a series of membrane channels. And then the rough endoplasmic reticulum is actually connected to the nuclear envelope. So it's connected to the nuclear envelope. It is this series of plasma membrane, folded plasma membrane channels, and then it has the ribosomes attached to them. And the reason that it's thought of as the factory is because then this is the site of protein synthesis. So why don't we go ahead and say the function then. Site of protein synthesis. So we have protein synthesis here, and these are going to be proteins that are going to be secreted by the body. So we have the free ribosomes that will be making proteins that can stay within the cell and provide cellular functions in the cell. And then when we go to the rough endoplasmic reticulum, when we meet up with that mRNA on a rough endoplasmic reticulum ribosome, then we're going to go through some further processing. And so the rough endoplasmic reticulum is actually going to have some further processing of proteins made by the ribosomes. So the ribosomes that are attached to the rough ER actually are then going to be pushed inside of the rough ER, inside of these folded units here. So if we have the amino acids that are all attached to each other, then we have our polypeptide that's going to be pushed inside of these little membrane channels. And then when it's pushed inside of the membrane channels, as it's going along in these membrane channels, it's actually going to be adjusted and changed, and we can move things around and add different things inside of the rough endoplasmic reticulum, also called the rough ER, so it can be shortened to that. Now the other endoplasmic reticulum is in this illustration over here. It's the smooth endoplasmic reticulum. They did put another rough endoplasmic reticulum over here that you can compare it to. The smooth endoplasmic reticulum and the rough endoplasmic reticulum. The reason this one's called the smooth endoplasmic reticulum is because it looks smooth. It looks smooth, of course, because it does not have the ribosomes on it. So our smooth endoplasmic reticulum has no ribosomes, and the function, so this would be like the structure. The structure, no ribosomes, and then they also have these membrane channels, right? So the same types of membrane channels, but just without ribosomes. And then the function of the smooth endoplasmic reticulum is that this is where we have lipid synthesis. So it's the site of lipid synthesis. So it's where we make our lipids. And this is also the site of detoxification. So we have in our liver, where we detoxify things, namely alcohol, which is a poison, and other types of drugs, are going to be detoxified within the cells of the liver. The cells of the liver then have high amounts of smooth endoplasmic reticulum because they're going through high amounts of detoxification in the body. So the next organelle that we're going to talk about is the Golgi apparatus, or the Golgi body. And we can see it's labeled here, the Golgi body, and it is up here. It looks very similar to our endoplasmic reticulum. So our rough ER and our smooth ER, where they have these flattened sacs. And that is exactly correct. So when we talk about the Golgi body, also called the Golgi Apparatus. We can think of this as the warehouse. This is the warehouse of the cell. The structure, as we just saw, very similar to the rough endoplasmic reticulum, the smooth endoplasmic reticulum. So a series of membrane sacs. So instead of channels where we have one continuous membrane that kind of flows in and out, making these channels, instead what we have are actual flattened sacs of plasma membrane that are somewhat separate from each other but sitting next to each other. They're all stacked on top of each other. And the function of the Golgi apparatus, or the Golgi body, is that it sorts and it modifies and it packages up proteins and lipids for secretion. And this is why it's considered the warehouse of the cell. So when our mRNA, messenger RNA, goes out to, let's say, the rough endoplasmic reticulum, it's going to meet up with a ribosome on there. The ribosome is going to help make the series of amino acids attached to each other, that first stage of making a protein, makes the polypeptide. That polypeptide will go into the rough endoplasmic reticulum. In the rough endoplasmic reticulum, it can kind of modify that polypeptide. It can make it go through the secondary and tertiary structures. And then it will be sent off via vesicle, which we'll talk about, to the Golgi apparatus. Once it gets there, we sort the proteins that are coming to the Golgi apparatus. We can modify them. We can add more polypeptide chains to make our finished protein. And then it's going to package those up and then send them out for release. So it sorts, modifies, and packages proteins and lipids coming from our smooth endoplasmic reticulum for secretion. So this is going to accept these proteins, the proteins and lipids, in vesicles, which are like little tiny bubbles that move around the cell, in vesicles from the ER. So again, proteins coming from the rough ER, lipids coming from the smooth ER, and then it releases them in vesicles. These are transport vesicles, and we'll talk more about those in just a moment. So our Golgi apparatus is going to work with our rough endoplasmic reticulum, which works with the ribosomes. It also works with our smooth endoplasmic reticulum to modify, sort, package up these things so that they can be secreted from the cell, so they can leave the cell. So we're going to talk now about these vesicles. So here, just kind of looking overall, we've already talked about the nucleus, remember, which stores DNA. When we talked about RNA, now we've talked about the rough endoplasmic reticulum with ribosomes, the smooth endoplasmic reticulum without ribosomes, sending that information, that stuff, to the Golgi apparatus. And we've talked about how these things are moving from place to place. So our RNA leaves the nucleus, and it can go to, our mRNA specifically goes to a ribosome, let's say on the rough endoplasmic reticulum. And then on the rough endoplasmic reticulum, we make these specific proteins. They go through these channels on the rough ER. Then they leave the rough ER via vesicles and then go to the Golgi apparatus for further modification and sorting. And then they'll leave the Golgi apparatus to be secreted from the cell. Now, all of this is done using vesicles. Our vesicles are like little trucks in the cell. So this right here is an example of a vesicle. So our vesicles are like our little trucks moving stuff around the cell. And this is because all of these things are made out of membranes. These are organelles. So again, remember, if we look at our rough endoplasmic reticulum, we have a protein that's been made in here. This part of our plasma membrane can just kind of separate off and can travel to the Golgi apparatus and glob on to the Golgi apparatus. Remember, our plasma membrane is fluid, so it can change shape and it moves around, so we can kind of grab some of it, move it around, and glob it onto other places that are also made out of plasma membrane. So let's take a closer look at the vesicle. So our vesicles are like our trucks in the cell. They're going to be moving things around. So the structure of them, the structure is that they are membrane-enclosed sacs that are going to bud off of the ER, the Golgi apparatus, or the plasma membrane. So, as we said, they are membrane-enclosed sacs that bud off the ER. So whether or not it's rough endoplasmic reticulum or smooth endoplasmic reticulum is just whether or not it's making proteins or making lipids and bringing them. The Golgi apparatus, and the Golgi body, which is also made up of plasma membrane, so it can bud off of the Golgi apparatus, or the plasma membrane. So they can then come from the plasma membrane. So when the plasma membrane is going to grab onto something and bring it into the cell, then it can be brought through the cell in a vesicle. Or we can send things out via the vesicles. So the function then of these vesicles are to store and transport material, and then they can also break down substances. Now, breaking down substances is just depending on if it contains particular enzymes. And we'll talk about that. So we have different types of vesicles. So we have our transport vesicles. And those are the ones that we're saying are going to be carrying materials from one part of the cell to another. From one part of the cell to another. So whether or not it's coming from the rough ER and bringing it to the Golgi, and then the Golgi to the edge of the cell, the plasma membrane for release, it's a transport vesicle that's moving it from one place to another. Now, when we're talking about those that are able to break down substances, we have a couple we'll talk about. So lysosomes are one that are able to break down substances. These are found in animal cells only. And lysosomes contain enzymes that digest most molecules. So they digest most molecules, meaning that if we need to break something down in the cell, say a protein didn't fold properly, then we can use our lysosomes to grab onto that protein, bring it inside of the lysosome vesicle, and digest that protein, breaking it up into parts, into those amino acids, and then sending those amino acids out so that they can be recycled and used over again. Also, lysosomes are useful for when we have a bacterial infection. So the cell, if it's infected with a bacteria, can take a lysosome and it can attach it to the vesicle that the bacteria is in, or whatever if it's not in a vesicle, it can attach to the bacteria, and it will start to digest the bacteria. The other type of vesicle that I want to talk about that contains enzymes is called the peroxisomes. Now, the peroxisomes are going to break things down as well. They contain enzymes. But they are going to break down toxins. So it can be like a bacterial toxin, for example. It can be drugs. Toxins could be alcohol toxins. Hydrogen. Of course, alcohol is mostly due to the smooth endoplasmic reticulum. Not hydrogen peroxide. Sorry, hydrogen peroxide. Hence the name peroxisomes. Helps break down hydrogen peroxide and helps with free radicals and other items like that. So our peroxisomes are going to help make sure that our cells stay healthy. The lysosomes do as well because they'll digest things like bacteria, but the peroxisomes are going to assist in making sure that we break down any chemical substances in the body, as well as hydrogen peroxide or free radicals. Those are both things that are made within our cells during regular cellular processes. So we have those to assist us. All right, so that is our vesicle, or vesicles, and you can actually see, now that we've talked about the different types of vesicles, that we do have a lysosome pictured here, which is similar to the vesicle we have indicated over here on the other side. So we've talked about much of the organelles that we see here inside of this cell. We did talk about the cytoplasm already previously. So the cytoplasm is pointing to this kind of area, and then inside here, this is this liquidy gel type substance that all of these other organelles are floating around in. And then also what's floating around in that are different things like nutrients, like glucose, for example, and ions or electrolytes like sodium and calcium and things like that. So the cytoplasm is there. We will talk about the centrioles, and we will talk about... we won't talk about the cell coat, but we will talk about the centrioles when we talk about some different cellular processes, rather than talking about that now. So the next organelle that we are going to talk about is called the cytoskeleton. It's not really pictured here, but we can see it as a skeleton, as a cell, and it would end up looking something like this throughout the cell. So we would have these different filaments, which we'll talk about, within the cell, that are giving structure to the cell and acting as kind of highways for these transport vesicles. So I'm going to kind of draw this in, cytoskeleton. So let's talk about the cytoskeleton then. So the functions of the cytoskeleton, one is that it maintains the cell shape. And you can kind of tell this by the name. Cyto is related to a cell, and then skeleton is like the skeleton, like we would think of for a body, like the bones of a cell. So they are going to maintain the cell shape, just like our bones maintain the shape of our body. So it maintains cell shape and structure. And this is really important in animal cells, because remember, in animal cells we don't have the cell wall, which is going to give structure to the entire plant, even if the cells are kind of shrunken, because maybe they don't have enough water at the time. In a human body or in an animal, we don't have that cell wall, so we don't have the ability to maintain our cell shape, with the exception of the cytoskeleton. So that's what we have, is the cytoskeleton. So the second function of the cytoskeleton is to provide strength to the cell. So remember that our plasma membrane is very fluid, and it can move and it can change shape. So it's kind of this moving, squishy, maybe you can think of it as like the outside of a balloon, like the rubber part of the balloon. If you blow up a balloon a little bit, not all the way to where it's taut, but you put some air in the balloon and then tie it off, you can kind of squish it and mold it and move it. And that's kind of like the plasma membrane of the cell. Now the cytoskeleton then is going to provide strength to the cell. And again, this is important in animal cells because we don't have that cell wall. So it's going to make it so that instead of being able to completely squish the balloon and pop it, we have some sort of skeleton inside there to help provide strength to the cell. Another function is that it allows movement of material inside of the cell. Inside of the cell. So the way that it allows for movement of material inside of the cell is it actually acts as kind of a transport highway. So we have these long filaments that we'll talk about, and these filaments can have little tiny machines walk along the filaments, carrying the vesicles from one place to the other. So we already talked about the vesicles and how they move from the endoplasmic reticulum to the Golgi apparatus, and so there will be these cytoskeleton filaments that will have these little tiny organic machines that are moving along, that are taking that vesicle and carrying the vesicle from the endoplasmic reticulum to the Golgi apparatus. So it's going to allow things to move within the cell, utilizing this as kind of like a highway system. And then lastly, that it allows for the movement of the cell completely, or as a whole. allows for movement of the cell itself. So rather than stuff inside of the cell, it's going to assist with what I had talked about previously with changing the shape of the cell. So globbing some of it out and filling that up with the cytosol. So we have the skeleton, and it's going to assist in that. So when we look at the structure, then, of the cytoskeleton, we have different types of filaments. So they are long filaments of protein. And there are 3 types. So the first type that we are going to look at are the microtubules. And the microtubules, these are long, really thick filaments. So we're going to actually talk about the 3 different types, and they are in order of size, or not in order of size, but our microtubules are the largest. I often think of microtubules as being big straws inside of the cell. They're very, very, very thick and tubular, as the name implies, microtubules. So long, thick filaments, and then they form a network from the nucleus toward the membrane. Let's see, from the nucleus toward the membrane. So the function of these microtubules, then, is that they are the ones that are allowing for movement inside and outside of the cell. So as I mentioned before, they kind of act like roads. So they act like roads for moving the cargo. So we have these little kind of machines that are going to walk along these tubules, and then they're carrying the vesicles with them. These are little motor proteins, so they act as roads for motor. Wow, that's not motor. For motor proteins that hold cargo and walk along the microtubules. So some of these motor proteins are kinesin and dynin. And so kind of an illustration of that is that we would have these tubules, and these tubules are actually made up of a whole bunch of proteins. So I'm just going to draw some of them, but there are a whole bunch of these proteins that are making up kind of this back and forth lines and lines and lines and lines, making up the tubule. And of course this goes all along the edge as well. So if you can think of this 3-dimensionally, we have all of these different proteins that are making up these tubes in here. So this would be like the inside, let's say. So we have these tubes. And then what we have are these motor proteins. And so we have these motor proteins that end up walking along the tubules, and then they're carrying the vesicles on top. And so they're walking along the tubules, carrying the vesicles. And so then, these are like little feet that are going to lob onto the next part, and then this one moves up, and then this one moves up, and then the other one moves up, just like we would think of feet. Little tiny proteins that are going to be moving along, the kinesin moving along, carrying our vesicle here. So not only do our microtubules allow for this movement throughout the cell or within the cell, act as these types of roads, but they also form flagella and cilia. So they form flagella and cilia. So flagella is a long whip-like tail. That's not where we need to go. Long whip-like tail. And then flagella, we actually see these a lot in bacteria and archaea. bacteria and archaea. But we also see them in animal cells, only one single animal cell, and that's the sperm cell. So it's a long whip-like tail, and so we'll have our individual cell, and then it'll have the tail on it, and that's the flagella, flagellum singular. And so our flagellum is going to be made up of these microtubules. So if we took a flagellum and we kind of sliced it in this direction and looked down it would look similar to this tube we've already talked about, but it has lots of them. So if we cut the flagellum itself, we'd actually see these tubes, but in this formation here. So we have microtubule, microtubule, microtubule. They end up being in groups of 2. Microtubules all around the outside here. It's supposed to be 9 and 2. 1, 2, 3, 4, 5, 6, 7, 8. 9 of those, and then 2 in the middle. And so then we have our flagellum here that is made up of several microtubules. Again, this is a slice, so we're kind of cutting a straw and then looking down the straw, for example. Now, when I say that, what I mean now is that this whole outside edge is the outside of the flagellum, the tail, and then now these individual tiny circles are individual microtubules, so little tiny straws within the flagellum that we've just cut. And then cilia. Cilia are like little tiny hairs, or short hair-like projections. And these short hair-like projections may move a cell, so they can move a cell, for example, like bacteria. So bacteria can be covered in cilia rather than a flagellum. It can be covered in bacteria and can use that cilia to move itself around, or, like in an animal, it can move material along a cell. And we already mentioned that when we talked about the respiratory tract. So we talked about inside of the respiratory tract, when we cough up some mucus, that's because we have cells in our respiratory tract that are bringing up the mucus because they have cilia covering those cells. So they're bringing that mucus up because of the movement of the cilia. So we're taking a look at our long filaments of protein, our 3 different types, and that kind of concludes our microtubules. So again, our microtubules are our largest ones. They're like big straws inside of our cell. The next one that we're going to take a look at are actually the microfilaments, which are the smaller ones, or smallest ones. So our microfilaments are short, thin filaments. And they form a network. Under the plasma membrane. So they can quickly assemble and disassemble so that we can change shape. So quickly assemble and disassemble to change shape. So the function of these microfilaments is going to be to support the cell and to change its shape. So support and change shape of cell. So we actually have these other microfilaments that are called myosin. So one kind of use we talked about in the microtubules, we talked about the kinesin and the dynin that are going to be these little motor proteins that are utilizing the microtubules. In this case, when we're talking about the microfilaments, we can think of myosin. Myosin is a type of microfilament that we find in muscle tissue. Specifically our skeletal muscle, you can think of it. When we think of our skeletal muscle, it is a motor protein. And this myosin is going to grab onto another protein called actin. and it's going to allow the contraction of the muscle. So it's making the size of the muscles change shape. It's going to shorten that cell, that muscle cell. So this is important in muscle contraction. And then the last type of filament that we have is our intermediate filaments. And our intermediate filaments, as their name implies, are the middle-sized filaments. And these are the most stable. So our small ones, our microfilaments, are going to easily break and change and move. And then our tubes are kind of in the inside there. And they can be taken apart and reformed. But then we have our intermediate filaments. And then these are going to form networks at the nucleus and at cellular junctions. And these are going to provide strength to the cell. So the function is to provide strength to the cell, and then to form this network at the nucleus and cell junctions, which we'll talk about next. So we see these intermediate filaments as providing strength because they're going to form this network around the nucleus, providing protection to the nucleus, as well as at cellular junctions. And we're going to talk about cellular junctions next, which means that we have concluded kind of our tour of the cell. We've concluded our organelles and our tour of the different organelles. So now what I'd like to do is I would like to talk a moment about cell junctions. But first, kind of to summarize all of these organelles here, we have our cytoskeleton and our plasma membrane. Our plasma membrane is the outer wall of the cell. It allows things to come in and come out. Our cytoskeleton is going to provide structure. We just talked about the three different types of filaments. Then we have in the middle, we have our nucleus. Remember, our nucleus consists of our nuclear envelope, which is the outer region here, protecting the DNA that's inside. Remember, inside we have DNA. That's where we make copies of our DNA. It's also where we make RNA. We can think of the three different types of RNA, but more specifically, messenger RNA is going to leave our nucleus, so we can send our messenger RNA, leave the nucleus, and head to a ribosome on the rough endoplasmic reticulum. In the rough endoplasmic reticulum, we have our protein synthesis, because remember, ribosomes are what are going to synthesize proteins. As it does that, the protein will go into the rough endoplasmic reticulum. It will move around in these channels, getting modified if needed. Then it will bud out of the rough endoplasmic reticulum in a little vesicle, travel along the cytoskeleton to the Golgi apparatus, where it'll glob onto the Golgi apparatus, and then the little protein will now be in these membranous sacs. Now it'll move between these membranous sacs as it's getting modified and sorted until it globs off here in another vesicle. And then it will use the cytoskeleton to then attach to the plasma membrane, and then the protein will be released out of the cell through merging of the vesicle with the plasma membrane. We also have our smooth endoplasmic reticulum over here that's making lipids. Again, we're going to bud off a vesicle. We can use our cytoskeleton to glob onto our Golgi apparatus, allow for our lipid to get into the Golgi apparatus, and then again do the same thing and secrete it outside of the cell. We have our mitochondria here, our little bean-shaped item that has its outer membrane, and then the inner membrane is important because this is where we have our ATP synthesis. We're going to make ATP in the powerhouse of the cell. So then, of course, we have our different types of vesicles. We already talked about our transport vesicles, but then we have our other vesicles like our lysosomes and our peroxisomes that are going to assist with breaking things down if needed and keeping the cell protected. I also didn't talk about the smooth endoplasmic reticulum as being a place of detoxification. So we make lipids there, send those to the Golgi apparatus, but then we also have lots of detoxification happening at the smooth endoplasmic reticulum. All right, so that kind of summarizes our organelles, and just very quickly, I would like to talk about cell junctions, since we mentioned those at the end. So cell junctions, the functions function of cell junctions is to hold cells together and then to allow interaction between the cells. So hold cells together and allow interaction between cells. The structure of these cell junctions is that we have different types of cell junctions that we're going to talk about. But in general, the structure is that we have these adhesion molecules that connect the cells and link them to the cytoskeleton. So adhesion molecules that connect cells and link to the cytoskeleton. And then we have the three main types that we'll talk about. The first type that we're going to talk about is adhering junctions. So when we talk about adhering junctions, they can also be called anchoring junctions. These are very strong junctions. So adhering or anchoring gives you an idea of how strong they are, and they hold cells together. So if we took a look at an adhering junction, I think what I'll do is I'll move this over a bit. If we have a cell, and we have another cell right next to it in a tissue, what we would see is that, actually I should probably separate these a little bit more so we can show that junction better. All right. So these would actually be very close to each other, but I'm giving a little bit of space for illustration. So what we end up having are these proteins on the side of the plasma membrane. So of course these are cells, so this is the plasma membrane, as we just discussed, the outer region of the cell. And then we have these proteins that are going to go in here and go out of the cell. These would be some of our integral proteins, and we see these on both sides of the cell junction. They're going to kind of attach to each other. They look kind of like puzzle pieces there, where they're going to grab onto each other. And then it's attached to the cytoskeleton in here. So attached to the cytoskeleton, and so then we have our cytoskeleton, which is of course throughout the cell here, throughout the cell, giving structure to the cell, anchoring things, and then we have our anchoring protein here, and then we have these proteins that are leaving the cell, going through the plasma membrane, that are attaching to each other. And if you can imagine, all of these little kind of teeth area in there, the puzzle pieces, are going to hold these very close together. So if this cell starts to move this way and this one moves the other way, they're going to hopefully not move very far because we have these adhering or anchoring junctions. Now these are important in tissues that experience lots of force, lots of movement. So important in tissues that experience force. So areas like the skin, for example. So our skin has lots of adhering junctions because as we're moving around, as we are kind of leaning on our arms or elbows or sitting on our knees or anything like that, we're moving our tissue around, we're sliding things around, and so we have these adhering junctions so we're not tearing our skin while we're doing it. Another example is we have these in our muscles. So as our muscles are contracting and moving, getting longer and bigger and everything, then we want to make sure we're not tearing those either. So those are adhering junctions, the first type that we'll talk about, which are our holding cells together. They're very strong junctions. The second type are called tight junctions. So our tight junctions are also holding cells together. So they hold cells together very, very tightly, so nothing leaks in between them. Hold cells together very tightly. So nothing gets between them. So no leaks. So we can think about it as in fluid. So I'm going to go over here and draw our tight junctions, for example. So if we have, again, our cell, then I'm going to put the other cell very, very close to it, because our tight junctions keep things very, very, very close. I often think about our tight junctions as sewing needles, sewing in the cell. So we'll often see them as like little stitches in the cell. And this is kind of hard to draw a three-dimensional shape, but if you think about this being kind of a three-dimensional cell here, and it's sewing. If you think of taking a thread and basically just sewing two cells together, those are our tight junctions. So these are important in the skin, so we don't let things in, and also in the digestive tract. So we don't want to allow things to get through that we don't want to get through. So then our last junction that we're going to talk about, third one, are gap junctions. So our gap junctions actually create openings between cells to share cytoplasm and signals. I'll drag this up a bit. So this is what is going to allow cells to communicate very, 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 very quickly. So allows cells to communicate very quickly. So if we draw a gap junction type of thing here, so one cell here, another cell here, what we actually see is that we have these integral proteins here, and they're actually tubes. So little tiny tubular type proteins going through there, and then it allows for something to go in, little tiny tunnels, very, very quickly move into the next cell. So it's still holding the two cells together, but it's also allowing for exchange of information between the two. So it can allow, again, cytoplasm to go through, but then also signals. So this is really important in heart muscle. So an area where we are sending electrical signals, and we want the entire heart to be, or different parts of the heart, but large areas of the heart to be pumping, to be contracting all at the same time. So in order to do that, we need to have messages sent very, very quickly between the cells in the heart, and we can do this through the gap junctions. So those are our three different types of cellular junctions that you should be familiar with: our adhering junctions, our tight junctions, and our gap junctions. One last thing that I do want to talk about before we move on to the next lecture on plasma membrane and such is the extracellular matrix. So the extracellular matrix, its function, or functions, is one is that it fills the space between cells. So sometimes cells are very far apart, and when they are very far apart, then we have this space to fill in between. So for example, bone. There are bone cells, but then between the bone cells is where we have what we actually kind of consider as bone. This is the actual extracellular matrix, which is kind of the bone part of the cell, which is the calcium and phosphorus deposits. So fills the space between cells, because they're sometimes very, very far apart. We have some other types of tissue that send communication signals or send things between them. And so we have lots of space between these things. Secondly, it gives cells a place to attach. So again, if we kind of think of bone cells, we have the bone actual cell hanging out in there, and it's surrounded by this very hard, what we would think of as bone, but of course bone actually includes all of these things. But it's kind of this crystalline extracellular matrix. So the bone cells are actually able to attach to this and hang out inside of little tiny spaces in there. Also, sometimes it contains nutrients. Again, contains nutrients, also contains wastes, and then the signals that are moving between cells. So our extracellular matrix, we can think of it sometimes as blood, for example, blood plasma. The liquidy part of the blood, that's not the cells, so the cells are floating around inside of the blood. The liquidy part of the blood contains the nutrients, so it contains things like oxygen, which we need, and the glucose that we need, and the sodium and other electrolytes. It also contains the wastes, so the carbon dioxide that we want to get rid of, the uric acid or the urea. So all of these things are floating around in our extracellular matrix of the blood, which is the blood plasma. Our fourth function is that it is important to the function of some organs. So important to the function of some organs. So for example, the skeletal system, or the skeleton, let's say, right? Because what makes up the majority of what we think of as bone, that makes it very hard and strong for our body, is this extracellular matrix, the actual crystalline structure, the calcium and the phosphorus that's deposited. Also things like cartilage. So cartilage that we think of like in our nose or in our ears, most of that is extracellular matrix, and then we have individual cells that are in little tiny spaces that are kind of far from each other. So the structure then of the extracellular matrix is typically a network of fibers and fluid secreted by the cells themselves. So when we think of the skeleton, for example, the actual bone cells are actually secreting this extracellular matrix. They're the ones that are making the hard part of the bone, the calcium and phosphorus deposits. They're secreting it. Same thing in our cartilage example. So the cells of the cartilage are actually secreting what we think of as cartilage, the kind of rubbery cartilage, kind of chunky but hard type of cartilage. So the structure then is this network of fibers and fluid, but it can be very different. So the fluid part of our extracellular matrix can be watery, as in our blood plasma is very watery. It can be like a gel substance, kind of like the cartilage, or rubbery, which is kind of like other types of cartilage, or it can be very, very hard, like bone. So that is going to sum up our eukaryotic cells, talking about all the different types of organelles, also our cell junctions, so connecting our cells to other cells, and then the extracellular matrix, so the stuff that's around the cells.